So today, the topic to be discussed is interferons. Interferons can be used for the antiviral therapy along with the antiviral drugs. So first, we will start what are interferons. Interferons are any of several related proteins that are produced by the body's cells as well as defensive response to viruses. It is very important modulators of the immune response available for therapeutic use. And this particular protein molecules, the glycoprotein molecules, has highly pleiotropic properties. So pleiotropic properties means a particular gene has multiple effects or multiple functions. So what are the properties? They can function as an immunomodulatory action, direct cytostatic action, direct cytotoxic action, anti-proliferative activity, as well as apoptotic effects they have. So British bacteriologist Alec Isaacs and Swiss microbiologist Jean Lindemann, they have isolated discovered first this particular type of molecule in 1957. And they performed an experiment using chicken cell. So they cultured chicken cell and found a substance that interfered with viral replication. And henceforth, they have termed this particular molecule as interference factors, latter which were termed as interferon. Then Nagano and Kojima, two Japanese scientists, they also independently discovered this soluble antiviral protein. So interferons are actually naturally occurring proteins and glycoproteins secreted by the host cell or the eukaryotic cell, better to say, in response to viral infections, tumors, and other biological inducers. Structurally, they are part of the helical cytokine family, which are characterized by an amino acid chain that is 145 to 166 amino acid long. And these interferons can only combat that they are not only combat only the viral infections, they can also combat bacterial and other parasitic infections and also inhibit cell division and promote the differentiation of cells. They can be produced by all vertebrate animals and possibly by some invertebrate as well. So what is the general action of interferon? So when a tissue cell that is of eukaryotic origin is infected by a virus or a bacteria or any pathogen parasite, it releases interferon. So then one eye is missing here, so please correct it. Then it diffuses to surrounding when it binds receptors on surface of the adjacent cells. And here you can see, so first when a virus particle has entered here into these cells, so interferon genes are there here in the host DNA. Interferons gene are there, that is the yellow highlighted portion of the eukaryotic DNA. So they will induce, they will synthesize, means they will go for transcription, and they will synthesize some mRNA. This mRNA will synthesize some proteins, and these are the interferon molecules. So these interferon molecules will come out from this infected cell, and then this particular molecules will be received or accepted by the surrounding cell receptor, surface receptor of the adjacent cell. This is the healthy cell, this is the this is the infected cell. So these are the interferons molecule that are received by the surface receptor of the adjacent non-infected cell, and then this particular protein will bind with and stimulates the cell to turn on genes for antiviral proteins. So now this molecule will come into the nucleus, bind with the double-stranded DNA of the host cell, induce some protein synthesis via mRNA formation or mRNA synthesis. So this molecule will inhibit or protect the cell from the infection of the virus particle. Here you can see the virus is again entering into this new cell, but this particular cell has some molecules, these black dots or black round shaped molecules. They are actually preventing or blocking virus reproduction here. So in this way, they can prevent the spread of virus throughout the body. Next, so induce a state which is so they can means induce the host cell so that they can inhibit viral replication, and antiviral activity is induced, and also it induces anti-proliferative as well as immunomodulatory activities. So it has some additional functions, induces by the interferons that resulted in their evaluation as an anti-cancer agent also. So what are the types of interferons? Most probably interferons are having three forms: alpha, beta, and gamma. They are the recognized form, but they can be classified into two types. One is a type 1, which includes interferon alpha, beta, delta, epsilon, kappa, tau, omega, etc. And in type 2, they consist of gamma subtype. So this division of interferons, or these types of interferons, is based on the type of cell that produces the interferon and the functional characteristics of the protein. So what are the type 1 interferons? So type 1 interferons bind to the same receptor. All types of type 1 interferons I have mentioned here. All the types of type 1 interferons. These are the types of type 1 interferons. So all this type of interferons, they will bind with the same receptor, and induced primarily in response to viral infections of a cell or eukaryotic cell. Interferon alpha and omega produced predominantly by the leukocytes. Interferon beta can be produced mostly, most kind of cells, but especially the fibroblasts, they are producing interferon beta. So how they are functioning actually from the fibroblast cell, how interferon beta induction is happening during viral infection. So it's a type 1 type of interferon production or induction. So pathogen associated molecular patterns, what is known as PAMS. Here you can see this pathogen associated molecular patterns. They are induced or recognition by the intracellular pattern recognition receptor that are known as PRR, intercellular pattern recognition receptors. Some are there. So leads to activation of the transcription factor. What is the transcription factor here? This is NF-kappa beta. And the second one is interferon regulatory factor 3 or 7, interferon regulatory factor 3 and interferon regulatory factor 7 here. These are the two transcription factors. And another one is there, that is AP1, that is not shown in this picture. So the cooperative action of these transcription factors is required for full activation of interferon beta promoters. Here is your interferon beta promoter here. So they will cumulatively, they will act, cooperative action of this transcription factor is essential for the activation of the IFN beta interferon beta promoter. Now IRF3 is phosphorylated here. This IRF3 is phosphorylated by this kinase. This is the kinase. This is called tank binding kinase, TBK, tank binding kinase, or other kind of kinases. So they will phosphorylate this. Okay, so they will phosphorylate this IRF3, and which in turn are activated by RIG1. This is RIG1, which is known as retinoic acid inducible gene 1. This is retinoic acid inducible gene 1. And here is this MDA5. This is melanoma differentiation associated gene 5. So these are, this is associated via IPS. IPS means interferon beta promoter stimulator 1. Interferon beta promoter stimulator 1. So in association with this IPS 1, so NF kappa beta, here you can see NF kappa beta is activated. And this particular pathway is known as PKR pathway, means protein kinase R pathway. So with this PKR pathway, interferon beta will be synthesized. Here you can see IFN alpha or beta will be synthesized, and it will come out. Here you can see it will come out, and it will be received by the adjacent cells. So that is how, via this transcription factor initiation, IFN beta promoter can be initiated or activated, and then their transcription will proceed. Next slide is on type 2 interferons. So type 2 interferons, there is one subtype, that is interferon gamma, and binds to its own unique receptor. So own unique receptor is there for interferon gamma, produced by two types of cells, that is known as T lymphocytes and natural killer cells, or NK cells. The main purpose of interferon gamma is to signal the immune system to respond to the infectious agent, or else in cancerous growth. Both the cases, it is just imposing the signal to the immune system. So in case of NK cells, a part of the innate immune response in NK cells, so activation not dependent on antigen recognition. But in case of T cells, T lymphocytes, so it is a part of the adaptive immune response. The NK cells are a part of innate immune response. The T cells, they are the part of adaptive immune response. So activation here in NK cells is independent of antigen recognition, but activation here in T cells, it is not independent of antigen recognition. It is antigen specific. Okay, so that is how the NK cells are stimulated. Here you can see through this picture how the NK cells here you can see how the NK cells and the macrophages, they are initiating. So the NK cells first, NK cells, they are exposed to some inflammatory cytokines, that is known as this tumor necrotic factor. And here you can see, here is your tumor necrotic factor alpha. So NK cells are stimulated first to produce produce interferon gamma. When exposed to these inflammatory cytokines, means you can see these inflammatory cytokines here, TNF alpha, tumor necrotic factor alpha, and interleukin 12 from the macrophage. So then they will induce or produce interferon gamma. In case of positive amplification loop, here, positive amplification loop, interferon gamma then stimulates the secretion of these two cytokines, inflammatory cytokines rather, TNF alpha and IL-12, interleukin 12. So Next, you will see that the distinct receptor and the downstream signaling. So what we have studied, the distinct receptor is there. So the distinct receptors and the downstream signaling that is initiated by ligand receptor interaction are responsible for the biologic effects of the individual type 1 or type 2 interferons. So here you can see how this infected cells that is getting stimulated, and and interferon alpha which or gamma that is coming, and here this particular pathway, one particular pathway, they are actually initiating this called the JAK-STAT pathway. We are I'm coming to that later. So here also you can see we have already discussed about this alpha and beta, the first one, and this is the gamma, this is the type 2. So here you can see the mitogenic antigenic stimulation that is happening here in IL-12 or interleukins, the cytokines, inflammatory cytokines, and then the IFN genes are there, which is in chromosome number 9 and chromosome number 12. 9 is alpha and beta, 12 is gamma. So they are actually activating interferons, releasing interferon, activating interferons genes and releasing interferons, and then it is received by some other gene, other cells, and they are combating this viral infection. So this is a scheme, a schematic summary of interferon system, how the interferons alpha, beta, and gamma, they are actually activating after the infection of by any kind of animal virus. So research conducted in 1970s revealed that these substances, this glycoprotein or protein substances, could not only prevent viral infection, but also suppress the growth of cancer cell. And they have proved this using some laboratory animals. So from there, hopes are raised that interferons might be the miracle molecule that might prove to be the wonder drug able to cure a wide variety of diseases. But its serious side effects, which include flu-like symptoms of fever and fatigue, as well as a decrease in the production of blood cells by the bone marrow, deflated expectations for its use against less serious diseases. So, despite these setbacks, in 1980s, alpha interferon came into use in low doses, very low doses, to treat mostly for different type of cancers. First, it was used to treat hairy cell leukemia, a rare form of blood cancer, and in high doses to combat Kaposi sarcoma, which frequently appeared in AIDS patients. So the alpha form also has been approved for treating the viral infections in case of hepatitis B, hepatitis C, and genital warts, which is known as condylomata acuminata, genital wart. The beta form of interferon is mildly effective in treating the relapsing remitting forms of multiple sclerosis. And the gamma interferons, that is used to treat chronic granulomatous disease, hereditary conditions in which white blood cells fail to kill bacteria. So interferons are categorized by cytokines, small proteins that are involved in intracellular signaling. Interferons is secreted by cells in response to stimulation by a virus or other foreign substances, but it does not directly inherit the virus's multiplication. So far we have studied, we are saying that it is just awaking the surrounding cells, that something has happened in your locality. So It has the function of awakening. It does not directly inhibit virus multiplication, but only awakening the surrounding cells. So what is actually doing? Rather, it stimulates the infected cells and those nearby to produce proteins that prevent the virus from replicating within them. Further production of the virus is thereby inhibited and the infection can be stopped. Means virus cannot proliferate in different type of cells or infect other cells throughout the body. So interferons also have immunoregulatory functions. So what are the functions? They inhibit B lymphocyte, the B cell activation, enhancing the T lymphocyte, the T cell activity, and increase the cellular destruction capability of natural killer cells. How they are doing this? What is their biologic effect? So here, interferons alpha and beta, as I have already mentioned, that they share the same receptor, which is composed of two subunits. The binding of either interferon alpha or beta to this receptor results in the activation of genus kinases, which is known as JAK1 and JAK2 genus kinases. Here you can see in this picture, here you can see this is the receptor. This is your JAK1. So it's a kinase, so phosphorylation will happen. So the signal which results in phosphorylation of transducers and activators of transcription 1 and 2. So here, which is the STAT1, the blue one, here you can see phosphorylation, and STAT2, the pink one, here you can see the phosphorylation. So STAT1 and STAT2, which is your phosphorylated form, results in their here in the cytoplasm, results in their heterodimerization. Here is your heterodimerization form. Dissociation from the interferon receptor. So these two, they are dissociating from here. Here you can see these are the interferon receptors. Now they are dissociating and just forming this heterodimer here into the cytoplasm. Now they will enter into the nucleus. So in nucleus, STAT complex, STAT1 and STAT2, they bind with the DNA protein P48. Here they will bind with the DNA protein P48. So this P48 is nothing but this is interferon stimulated gene factor 3, ISGF3, which binds to the interferon stimulated response element. Here you can see interferon stimulated response element of the alpha and beta responsive gene, here of the alpha and beta responsive gene. An induction of the interferon targeting site gene or interferon target genes responsible for the biologic effect of interferons alpha and beta. So that is how the biologic effects of interferon alpha and beta is carrying from the cell membrane to cytosol to the nucleus, and they are actually stimulating the complex, or they are actually inducing the interferon target genes responsible for the biologic effects of interferon alpha and interferon beta. Next one is the biologic effect of interferon gamma. So interferon gamma will bind with different type of receptor. Here you can see it's a homo receptor, a homodimer. Here you can see the homodimer, IFN GR1, the homodimer, specific interferon gamma receptor. So this is a specific interferon gamma receptor, and this is the homodimer IFN gamma, IFN gamma. So dimerization of the receptor activates again genus kinase 1 and 2, JAK1 and JAK2, which ultimately results in the phosphorylation of STAT proteins stat 1 and stat 2. So here also stat 1, this blue one, and this blue one you can see. Now the stat 1, instead of activating stat 1 and stat 2, so what they are doing, interferon gamma activate two stat 1 protein, two stat 1 molecule. Means there is, in the first case, alpha and beta, that is heterodimer, and here we are getting the homodimer. There the dimerization between stat 1 and stat 2. Here the dimerization between two stat 1 molecule. So this is a homodimer, not a heterodimer in case of interferon gamma. So they form a homodimer known as gamma activated factor, which is known as this one. This one is known as GAF, GAF, gamma activated factor. And these translocate to the nucleus and binds to the gamma activating sequence, which is known as gas sequence, gamma activating sequence. Elements of interferon gamma inducible genes. So this is the element of gamma inducible interferon gamma inducible genes. So now this will induce production of or synthesis of interferon gamma, and it will be released from the infected cells. So this antiviral activity of interferons is mediated by three pathways. What are the pathways? One is 2' 5' oligoadenylate synthetase pathway. The second one is double-stranded RNA dependent protein kinase R pathway, and the second one is MX pathway. So first one is 2' 5' oligoadenylate synthetase pathway. Here first ATP, adenosine triphosphate, will go for oligomer formation. Then it will bind with ribonuclease L, cleave single-strand RNA, and henceforth inhibition of protein synthesis and cellular proliferation will be inhibited. This is a bit simple pattern. The second one is double-stranded RNA-dependent PKR pathway, protein kinase R pathway. Here, this PKR is activated by binding to double-stranded RNA. So here you can see PKR binding with double-stranded RNA. Once activated, PKR phosphorylated. Here you can see the phosphate. Phosphorylates the translation initiation factor. This one is the translation initiation factor, EIF2, which inhibits, this one is inhibiting mRNA translation, messenger RNA translation. So no protein will be synthesized. And PKR, in the second way, they are actually activating NF kappa beta. So the first method we have discussed. Please recall the first figure we have discussed. So this PKR is activating this transcription factor NF kappa beta, which can lead to increased production of chemokines and cytokines. Maybe inflammatory cytokines, chemokines. So chemokines level, cytokines level will be high. So this increased PKR activity can also induce apoptosis by BCL2 and caspase dependent mechanism, means mitochondria mediated pathway. Now the third one, that is the MX pathway. So another group of protein induced by interferons that exhibit antiviral properties. This is the MX family of glutamine transpeptidase. So MXA is a particular protein produced during viral infections and inhibits viral replication at the level of transcription by binding to susceptible viral nucleocapsid in the cytoplasm and preventing their movement into the nucleus. So they are not allowing the genetic material to the host cell nucleus. So by inhibiting this step, they are preventing their transcription, viral DNA or RNA transcription, and also they are inhibiting the formation of nucleocapsid. Now, some oncologic applications of interferons. So, interferons alpha N3, which is also known as alpharon N, that can be used for genital and perianal warts treatment. Interferon beta 1b, which is also known as beta seron, and beta 1a, which is the name is Avonex, can be used for multiple sclerosis. Interferon gamma 1b can be used for malignant osteoporosis, osteoporosis. Interferon alpha 2b for chronic hepatitis C and hepatitis B. In case of some tumors, established indications is there: chronic myelogenous leukemia, hairy cell leukemia, non-Hodgkin lymphoma, acquired immune deficiency related to Kaposi sarcoma, malignant melanoma, or else we can use for retinal cell carcinoma, bladder carcinoma. So these are the tumors, these are the type of cancers where interferon therapy can be used. But interferon therapy has some organ toxicity, mostly hepatic toxicity, acute hepatic toxicity. And this acute hepatic toxicity can be manifested as increase in serum level of SGOT or SGPT. It can be fatal. Fatal complications can be avoided with careful monitoring and appropriate dose modifications of interferon, different type of interferons.